Maria José Alonso, professeur Alonso, bienvenue et c'est à vous tout de suite pour votre conférence.
Mr. President Kraman, Mr. New Elected President Casimir, members of the Academy, authorities, ladies and gentlemen, it is a great honor for me to be here today to become a new member of the Academy, Royal Academy of Medicine of Belgium. It is a great honor, and I want to thank the members who nominated me to become this foreign member of this academy. So let me start by presenting you my research team, who is in front of our research institute, SIMUS Research Institute. It's an institute on molecular medicine, and so we are all working there. I'm also professor of the Faculty of Pharmacy at the University of Santiago de Compostela, and I have also been the Vice Rector of Research and Innovation of this university. So my career started at the University of Santiago de Compostela during my PhD, and then it continued in the University of Paris Sud, also at the Institute of Gustave Roussy in France, and at the University of Angers, and finally at the Massachusetts Institute of Technology, MIT, and then I came back to Spain. So just to show you, who I have been learning from, Professor Beila Hato was my PhD advisor, and I was working on increasing the bioavailability of drugs. And then with Patrick Couvreur, actually a member of the Academy, I learned making nanoparticles and starting in the field of nanomedicine, in particular for the treatment of infectious diseases. At that time, I also worked with Antoine Andremont, l'Institut Gustave Roussy, and also with Jean-Pierre Benoit, who is at the University of Angers. Finally, I went to the US, where I did some work on vaccine delivery with Professor Robert Langer, and have spent many times in this Institute of Technology. And from that time until now, I have always been working on nanoparticles for delivering drugs or vaccines. So these are, the main goal has always been to help biological drugs or antigens overcoming biological barriers. And my major accomplishments in research, I would say, could be classified in 3 lines: global vaccines using vaccine delivery, development of nanocarriers for overcoming mucosal barriers, and personalized nanomedicine in cancer. A few words about each of these areas. So global vaccines, we had a chance to participate in the Grand Challenges in Global Health program of the Bill and Melinda Gates Foundation, and also in collaboration with the World Health Organization. Our approach was to design single-dose vaccination strategies and also needle-free vaccination approaches through the use of the micro and nanotechnologies. Our goal was also to develop thermostable vaccines. Up until now, we have been working with a great variety of antigens, starting with tetanus toxoid and with hepatitis B, E. coli. Over the last decade, we have dedicated all our efforts to a new HIV vaccine in collaboration with the US and Canada. And actually, this vaccine is in the late phase of preclinical development. So as you can imagine, working for 30 years on drug delivery, on the design of nanoparticles for delivering these vaccines, we have made many different kinds of structures, as you can see in this slide. And if you think about it, they are very similar to the structure of the virus, the coronavirus in particular, in the sense that we have RNA inside, for example, here, and then a number of components, lipids, polysaccharides, and proteins. So we have been working almost for 1 year now, starting on March last year, on the development of a new COVID vaccine, actually in collaboration with Belgium, several groups from Spain and Belgium. And basically, this is our mRNA vaccine. The name of the consortium is Covarna. So in Belgium, Chris Tillman fabricated the mRNA fragments that we incorporated in our nanocapsules. These nanocapsules enter the cells and deliver the RNA inside, helping the production of the antigen inside the cell. So once we had these spike protein antigens, they will be able to develop the required response, IgG response, and T-cell response. And this is where we are now. Other areas of activity where we have, would say, really done some milestone work is in the field of nanocarriers for overcoming mucosal barriers. In particular, we have been dealing with the intestinal barrier and with the nasal mucosa. So regarding the oral modality of administration, we have used these nanoparticles indicated here for transporting drugs. This is a belly of the GI tract for transporting drugs such as insulin across this epithelium. Regarding the nasal modality of administration, currently we are developing new technologies that enable the transport of these particles across the nasal mucosa and even to reach the brain for the delivery of RNA. And finally, regarding our work on personalized nanomedicine in cancer, we have developed a technology, multifunctional polymeric nanocapsules, that enable the intracellular delivery of monoclonal antibodies. And this is a paradigm shift in the sense that up until now, all the monoclonal antibodies marketed, especially for the treatment of cancer, are only intended to reach extracellular targets, while there are many intracellular targets that remain undruggable. So we have incorporated monoclonal antibodies within nanoparticles that enable the transport of the antibody towards the intracellular target. Therefore, this is a new concept, therapeutic antibodies against intracellular oncoproteins, that allow us to drug some known as undruggable targets. And these are the particles going into the cells and delivering inside the cells the monoclonal antibody. This is a real image of the nanoparticles, and this is the appearance of the liquid suspension of them. So just a couple of photos of my research lab, and then just to thank you as a final slide, just to thank Veronique Pret for having the initiative of presenting me to the Academy. Of course, I also thank my parents, my nominators, my mentors, my fellows, my friends, my family, my university, and all supporting institutions. And I finished my presentation with these beautiful flowers, Hortensia and Camellia, very typical from Galicia. Thank you very much.